Hello, everyone. This is my first time at OIS and first time on the stage, so hopefully I can be clear to you. So I think at Ingenia, we are developing a therapeutic antibody, transforming the dysfunctional blood vessel into healthy blood vessel, indicated by the gray and the red areas in the logo. So we are targeting microvascular disease, currently focusing on the ophthalmic indication. So we are founded in the strong science. We have successfully assembled talented and committed research staffs and experienced management. And also we have a renowned advisory teams. And we started a collaboration with the Mosaic Bioscience to develop the site-specific pegylation technology. We believe we have a strong ophthalmology pipeline. So we have two molecules. The lead program called IH427 is targeting DME at AMD. And also the other molecule is targeting glaucoma. With the well-prepared resources and roadmap, we are targeting to file the first clinical trial the first half of 2024. So let me talk about the main idea for our lead program, which is a bispecific antibody and pegylated molecule. So in addition to VEGF blocking, our lead program is activating Tie2, which is a well-established receptor tyrosine kinase, which is playing a pivotal role in maintaining the preventing the vascular leakage and controlling the responsiveness to VEGF. For the NG2 inhibition NG2 inhibition approach, by capturing neutralizing angiopoietin 2, the locally available angiopoietin 1 is binding and activating Tie2. But the degree and magnitude of the Tie2 activation is, in this case, is dependent on the locally availability of angiopoietin 1. The level of angiopoietin varies depending on the stage of the disease progression and also the patients. And in many cases, the level of angiopoietin tends to be very low. So the simple NG2 blocking might not be enough to drive the full-blown activation and optimal activation of Tie2. So in contrast, what you found using our molecule, by directly activating Tie2, we can provide a more robust and durable activation of Type2, regardless of the concentration of angiopoietin 1, angiopoietin 2. We also found that our molecule can have a very unique feature. It can block the Tie2 cleavage from the protease, which is elevated in multiple disease conditions. So with that, we believe that a stronger and durable Tie2 activation might be possible by direct activation of our molecule, and it can better stabilize retinal vessels beyond the standard of care. So here's how our molecule looks like. This is a bispecific antibody with a site-specific pegylation. 4 features. One, it has a dual function, VEGF blocking and also Tie2 activation. The other is it has a block and prevent the activity toward Tie2 proteolytic inactivation. It drives the strong and durable Tie2 activation. And it also, via the site-specific pegylation, it has a longer VTR half-life. With that, we believe that IH427 is specifically designed to provide improved tolerability and retinal health. Let me give you some key slides, key data about this molecule. So we found that IH427 can trigger the strong and durable Tie2 activation compared to the endogenous Tie2 agonist ligand called angiopoietin 1. And we also found that through the rapid ocular PK study, pegylated version of the 427, it has a doubled longer the ocular half-life compared to the unpegylated version of our molecule and also other SOC competitors. So with that, I think our molecule can provide a robust and durable Tie2 activation, which can lead to the better pharmacodynamic effect and reduce injection frequency. The other feature which I found was very unique for our antibody, it can block the Tie2 cleavage and shedding. in normal condition or a pathologic condition, it is well understood that Tie2 is cleaved by the in many conditions, including angiogenic or inflammatory hypoxic conditions. These conditions is leading to the upregulation of protease, which is leading to the Tie2 cleavage and shedding. So in the end, the soluble cleaved Tie2, called soluble Tie2, from its production is increased and circulating in our body, which is leading to the destabilization of vasculatures. So what is a unique thing from the structural study, what we found that our molecule's epitope overlaps with the MMP-14 cleavage site. MMP-14 is one of the dominant proteins found in these disease conditions. So what we found, our molecule can block the cleavage of Tie2 from the MMP-14, and then our molecule can inhibit the production of the soluble Tie2. So this is adding another layer for the Tie2 production from our antibody. So it can preserve and protect Tie2 in our body, including normal and disease conditions. So that is a very unique feature of our molecule. And recently we created a kinetic model based on the pharmacokinetic parameters and pharmacological activity constants to predict the durability of the VEGF inhibition and Tie2 activation compared for our IgG4 variant along with the competitive molecules as a standard of care. So as you see here, this model demonstrates a clear increase in the level of Tie2 activation and also at the VEGF blocking in a time-dependent manner. As you see here in this table, there's a beautiful increase of all the duration in the Tie2 activation and VEGF trafficking. So trapping. So we think that this is a quite compelling model which can provide a less frequent injection compared to the other competitors as a standard of care. So as a summary, we will IgG427 is uniquely designed for the improved clinical outcomes. It has multiple functions, and a very unique and differentiated mode of action, as I told you, including the blocking the VEGF and activation Tie2. It also preventing Tie2 cleavage and loss, and has a longer half-life. So based on that, we believe that our molecule can provide better efficacy and safety, along with the analytical response and durability, which is leading to the exciting hypothesis that our molecule can provide a very compelling alternative to the patient and can cover the broader patient population. Thank you for your attention.